Thank you for this opportunity and organizing, Dr. Noel. Age-related retinal degeneration, ARD, arises from failure of tissue maintenance and repair pathway that were accelerated by inherited and acquired factors. Although potential markers have been identified for ARD, however, the set of genes which can be accelerated or triggered ARD and makes ARD to a chronic state is still unclear. To answer this question, so we investigate a genetic background of AJ and the basic two different kind of inbred mouse model, which diverse progression of ARD. So retinal cross-section of AJ at 8 months old pronounced a severe photoreceptor cell death, and simultaneously pathological change of RP cells, contrast to 1 month old of AJ here. Conversely, B6 mouse showed very mild age-dependent changes at 8 months old compared with a 4 weeks old mouse. Fundus images obtained by scanning laser ophthalmoscopy. This is an in vivo imaging system. So we can see numerous number of bright spots on the 8 months old AJ mouse in a fundus, whereas we can't see any such a kind of fundus, fluorescent spot in the 4 weeks old of age. So these pattern of these fluorescent spots can be inflammatory cells. Potential inflammatory cells invasion is observed by a high definition SDOCT system in the subretinal space, and in the plastic section we can see the invasion of potential these cells in the subretinal space and the outer nuclear layers as well. These cells are identified by immunohistochemistry using specific antibodies for activated microglia and macrophages, EVA1 antibodies. And also, GFAP activated mirror cells are positive for 8 months old AJ mouse. It's not positive in 4 weeks old and 8 months old B6 mouse. So from these observations, inflammatory event involves for ARD pathology, and also this ARD AJ mouse retina is under stress condition. To investigate this genetic background for this mouse, so we conducted 2 unique genetic dissections for these studies. So first one is using genetic dissection using with chromosome substitution strains. Each CSS contains 1 substitute chromosome which is donated from AJ. So for example, so A1 strain has only 1 chromosome, only chromosome 1 donated from AJ. And for example, A12 carried chromosome 12, which is donated to AJ. So from using these CSS strains, we can analyze specific phenotypes coded in each chromosomes here. So we focused on 2 factors for this CSS. So one is 11-cis retinal visual chromopher. So this amount is represent visual pigment amount in the retina. And second one is a number of nuclei which attribute to a photoreceptor cell death. So very interestingly, no CSS can represent severe retinal degeneration, which is observed like us in AJ mouse. However, so we can see very subtle differences in between the CSS. So from these observations, so we can speculate that this this severe ARD observed in AJ is induced by many different genetic factors located in different various chromosomes. To identify multi-genetic factors, so we conducted deep RNA sequencing using Illumina system. So this analysis is conducted by super graduate student Devaraj. So we did more than 13,000 transcriptome area, and this RNA sequences provide us a very accurate determination of the genetic profile between AJ and B6 mouse. Actually, so we can see very strong correlation with RT-PCR at 4 weeks old and 8 months old between AJ and B6 mouse. And also, this profile is determined by a Western blot for retina-specific anti-retina-specific proteins. So this method is really practical to analyze the background of these diseases. And we picked up 3 genes important for tissue maintenance: GPX3 and Corin3 and MYO7A. The function of this encoded protein GPX3 is hydrogen, so fighting to a hydrogen peroxide. So this protein function is hydrogen peroxide detoxification. It means that GPX3 is fighting with oxidative stresses. And MYO7A and Corin3, that two protein has an overlap the function. So MYO7A's function is a protein and phagosome trafficking, and Corin3 is function is endosomal and acidification and formation. Immunohistochemistry using specific antibodies for this protein is positive in the marginal expression in RP cells marginal expression in RP cells. However, this signal is severely attenuated in the AJ mouse. Given this data, so we look at we try to get more detailed information in RP cells. This is a high definition EM pictures in a cross section of RP cells. So we notice that inclusion body in the upper layer of RP cells. To get better images for RP cells, we prepared horizontal sectioning of these RP cells. So what we found is numerous number of vacuole formation in RP cells, which is not observed in B6 mouse and 3 months old. So at 3 months old, so I think so. We think that already pathological changes occurred in RP cells in the AJ mouse and induce precipitate and induce age-related retinal degeneration. When we look at across this vacuole formation, so in case of AJ mouse, we can see a double pocket, and the inside is filled with undigested outer segment of photoreceptor cells. And this pocket is broken, and this inside of contents is exposed to endoplasmic space in the RP cells. However, in case of a B6 mouse, so we can see very compacted, nice formed phagosomes in the RP cell, outer segment from photoreceptor cells. And this phagosome is transported to the dorsal side, the apical side of RP cells. It means this digestion is properly processed. On the other hand, the AJ mouse, so this phagosome is still in the surface of RP. These pathological changes observed by a 2-photon microscope as well. So from 4 weeks old to 8 months old. In case of 4 weeks old, so RP cells size and the morphology is quite healthily maintained. However, it's changed morphology is changed dramatically from to 3 months to 8 months. So now 8 months, the morphology is completely disrupted, and the size is increased more than 1.5 times, and so we can see very swollen, sick-looking RP cells in the AJ mouse at 8 months old. On the other hand, so B6 mice, so function and morphology and is quite well maintained in the B6 mouse. From these observations, so these downregulated tissue maintenance genes are affected to a function of the function of RP cells, which is the trigger factors to induce the ALD. In fact, we can see a significant reactive oxygen production in the RP cell layer in the AJ mouse at 8 months old, and this signal is enhanced by light exposure. On the other hand, we can see such a kind of increase of loss production in the B6 mouse. More importantly, ingenuity pathway analysis revealed an activated inflammatory priming pathway in the AJ mouse at early age. So on the center of INF beta. So at that pathway is increased, evidenced by increasing of inflammatory products such as IRF7, STAT1, and the GBP1. And the more important thing is this kind of signal is increased by aging, so compared with the over-month old AJ mouse. So these factors involved and combined with the downregulated tissue maintenance factors, this is induce the severe type of age-related retinal degeneration in the AJ mouse. So summary of this study is so ARD, age-related retinal degeneration in AJ mouse was triggered by inflammatory priming coupled with absence of the set of key homeostatic molecules and age-related increase of inflammatory factors contribute to the rapid progression of ARD. And phenotype and genotype characterization using a combination of RNA deep sequencing and advanced imaging systems allows a more comprehensive understanding of a complex neurodegeneration such as ARD. So this slide is most important for my presentation. So I really appreciate, especially thanks to Dr. Palczewski and Debash for intensive support for this study, and Debash contribution for deep RNA sequences. I really appreciate. And Satsumi and Melissa for CSS characterization, and Brian and other Palczewski members, and from genetic departments, Dr. Nada and Ani, and the EM co-facility for wonderful EM imaging, and the project for technical support for two-photon microscope, and the visual support, and our laboratories. I really appreciate. I will stop my presentation. I'm happy to answer my question. Thank you very much.